So let's continue with SOS response. Previously we had mentioned upon the correlation between LEX-A protein and REC-A protein and SOS box. It has been found that the SOS box has been repressed by the action of LEX-A protein, and when DNA damage happens in an extensive phase, REC-A protein is being formed, and REC-A protein carries out the proteolysis of the LEX-A protein, thereby triggering or switching on the SOS response. So what is the role of REC-A? So as we told just now, the action of the SOS response, it mainly depends upon the REC-A protein. The REC-A, it usually binds to the single as well as the double stranded breaks as well as gaps which have been generated during the cessation of DNA synthesis. And it also initiates the process of recombinational repair, and I think I don't have to explain this because we have spent different sessions on the recombinational repair. And as we have mentioned earlier in the previous slide, you can see that REC-A also has a coprotease function, and what it does, it interacts with LEX-As and causing the LEX-A to destroy itself by a autoproteolytic reaction. When LEX-A is being destroyed, it also increases the transcription of various other genes which have been involved in the excision repair as well as in the recombinational repair. Now just have a look at this slide. It's the repetition of what we have told earlier, but you can see you have your damaged DNA. REC-A is forming, REC-A acts as a protease, and LEX-A is being it has been lysed, and as a result the LEX-A inactivation, usually if LEX-A protein is there, it will inactivate various genes, and some genes will be inactive normally. But when the DNA damage is there, various proteins like REC-A gets activated and UBRA gets activated and UMUA gets activated. So this is the cycle of the events which normally happens. When LEX-A is being found to be formed, when LEX-A is formed, what happens? The SOS is being inhibited, and when REC-A is being formed, the SOS is triggered. You just remember these three genes which are necessary for the SOS response. So the first genes transcribed in the SOS response are those that encodes the UVR proteins needed for the nucleotide excision repair. And then the expression of genes which have been involved in the recombinational repair, it is further increased. And you can also see that to give the cell more time to repair its DNA, sometimes the protein SFIA is also being produced, because SFIA will block the cell division, and even during that time, if the DNA is not being fully repaired, even after 40 minutes, what happens? Another process called the translation DNA synthesis is being triggered by the SOS response. So you have, in the first response, you have the UBR, which is mainly involved in the UBR genes have been triggered as a part of SOS response, and they are mainly responsible for the nucleotide excision repair. And so after that, more amount of REC-A is being triggered once the SOS response is being initiated, and that will result in the recombinational repair. And another one, if these two systems are not been working properly, and if that is not able to give sufficient repair even after 40 minutes, another system called the translation synthesis, DNA synthesis will happen, and that is mainly being initiated by a product of this gene, UMUA gene. Let's take a look onto that. In the translation synthesis, DNA polymerase 4, also known as the DIN-B, and DNA polymerase 5, UMU, okay, they are being synthesized, they will synthesize DNA across the gaps and other lesions. that has been stopped by the DNA polymerase 3. However, because an intact template does not exist, these DNA polymerases often insert incorrect bases, and they also lack the proofreading activity. Therefore, even though the DNA synthesis continues, it is highly error prone, resulting in the generation of numerous mutations. So, as a last response, when the first ones, that is nucleotide excision repair as well as the recombinational repair, is not sufficient enough to fill in the gaps or to overcome the damage which has been caused by the high exposure to UV rays, the translation synthesis will be initiated. And mainly two main enzymes called the DNA polymerase 4 as well as the DNA polymerase 5 will be synthesized during this time, during the translation synthesis. Now, what is a translation synthesis? That is, across the DNA which is having gaps in various other lesions. Lesions means, for example, thymine dimers might occur as some lesions. In such areas, what happens? The DNA polymerase 3 will not be able to synthesize DNA. And so, because the template has been highly damaged. In such a situation, DNA polymerase 3 becomes highly confused and it is not able to add further DNA molecules into the strand. So, in such a situations, when DNA polymerase 3 is not working, the SOS response triggers two other enzymes called the DNA polymerase 4 and DNA polymerase 5, and they will act and they will synthesize new DNA nucleotides into the DNA. But since the template is not there and since the proofreading activity is not there, SOS response will result in the what? It will result to error prone DNA synthesis. And usually it is named so because it is made in a life or a death situation, and the SOS response increases the likelihood that the cells will survive by allowing DNA synthesis to continue. And for the cell, the risk of dying because of a failure to replicate DNA is greater than the risk posed by the mutations generated by this error-prone synthesis. SOS is a saving response. So, rather than dying, the cell would prefer that, okay, let it be a error-prone DNA synthesis. I hope you got an idea of SOS response, which is a last response of a last resort of DNA repair system which has been followed by the microorganism. It could be due to high lethal damages caused by the UV exposure. When the photoreactivation system is not working, when that is not sufficient enough to overcome it, the SOS response will be triggered. With this, we come to an end of the SOS response. Thank you for your attention.